You are 56 years old, and you have been doing everything the breakfast magazines told you to do. You pour a bowl of quick oats almost every morning. You add a spoonful of honey, splash of milk, maybe a handful of berries if it is a thoughtful day. On paper, you are winning at breakfast. The packet says whole grain. The label says heart healthy. Your doctor has told you, more than once, that oatmeal is the one breakfast you cannot get wrong. And yet, almost no part of that statement is biologically accurate. Not for the oatmeal you are eating. Not for the effect it is having on your cholesterol right now. Not for what it is doing to your blood sugar in the 90 minutes after you finish the bowl. And definitely not for the one molecule sitting inside those oats that nobody on the label ever mentions. There is a strange pattern that shows up again and again in the research on this specific breakfast, and almost nobody explains it properly. Two adults walk into a doctor's office with the same LDL cholesterol, the same fasting glucose, the same waist measurement. 12 weeks later, after one of them adds a daily bowl of oats and the other does not, the cholesterol numbers have moved apart in a way the literature keeps reporting, without most news coverage ever picking it up. The difference is not the oats. Both adults think they are eating oats. The difference is the kind of oats. The difference is what was done to the oats between the field and the packet. The difference is what the bowl actually contains versus what the label claims it contains. So, what actually separated them? The answer keeps circling back to one molecule, almost nobody is told to look for. One number on the side of the packet that most adults ignore, and one mistake the breakfast industry has trained an entire generation to make at the convenience store shelf. And once you understand why it works, you will never look at the bowl on your kitchen counter the same way again. Before we go further, if you have ever wondered why some breakfasts quietly lower cholesterol, while other breakfasts quietly raise it, this channel breaks down exactly this kind of hidden biology every week. So take a second and subscribe now, because what comes next is going to reshape how you think about mornings, fiber, and the decades of small arterial damage the average adult is walking around with right now. Let us start with what is actually in the bowl. So what is oatmeal, exactly? The relevant grain is Avena sativa, a member of the grass family cultivated for at least 3,000 years, originally a weed in wheat and barley fields, before selective breeding made it its own crop. The relevant structure is the oat groat, the dehusked kernel that contains three things the breakfast aisle cares about: a starch reserve, a storage protein, mostly the globulin fraction, which behaves differently from the gluten of wheat, and a soluble fiber your small intestine cannot digest called beta-glucan, which is the molecule most of the clinical trial results actually sit on top of. Every form of oatmeal you can buy, steel-cut, rolled, instant quick cooking, Scottish, Irish, is just the same groat sliced, steamed, pressed, or pre-cooked to a different degree. And that slicing is what changes almost everything about how the bowl behaves inside the body. Steel-cut oats are simply the groat chopped into 2 or 3 pieces. They cook in roughly 25 minutes. Rolled oats are the groat steamed and flattened. They cook in roughly 5 minutes. Instant oats are the groat steamed further, pressed thinner, often partially pre-gelatinized, and cut smaller. They cook in 90 seconds. Take a moment to remember that sequence, because the entire health story we are about to walk through hangs on it. A cooked oat is not the same carbohydrate regardless of how it is processed. A cooked oat is a fundamentally different molecule depending on whether the starch was preserved in its tight crystalline form or whether that form was already broken open in the factory. Now let us connect this directly to the molecule the clinical trials keep measuring. The relevant molecule is beta-glucan, a soluble fiber made of long chains of glucose units linked in a beta-1,3 and beta-1,4 pattern. Beta-glucan exists in barley and in oats. In oats, it sits in the cell walls of the endosperm, specially concentrated in the outer layers of the groat. Its chemistry is unusual because the human small intestine cannot break the beta linkages. So the molecule passes through the stomach and the small intestine intact and arrives at the colon as a polymeric substance, your gut bacteria, and only your gut bacteria know how to metabolize. Why does this matter for cholesterol? Because along the way through the small intestine, beta-glucan does something else first. It forms a viscous gel in the lumen of the duodenum. That gel physically binds bile acids, which are cholesterol derivatives your liver secretes to emulsify dietary fat. The more bile acids the gel captures, the more bile acids escape reabsorption in the ileum and are excreted in stool. Your liver, in response, pulls circulating LDL cholesterol out of the bloodstream to manufacture new bile acids. That pull is the LDL drop the literature has measured across 2 decades. The Whitehead meta-analysis pooled the studies and reported a roughly 5%, or around 0.25 millimoles per liter, reduction in LDL cholesterol when adults consume at least 3 grams of oat beta-glucan per day. That is the dose. That is the threshold. That is the first number on the side of the packet most adults get wrong, because most adults assume that any oatmeal is oatmeal, and most adults do not weigh out 3 grams of beta-glucan to confirm the bowl contains it. Now, here is the part almost nobody explains properly. The viscosity of beta-glucan is dose-dependent, and the viscosity is what physically traps the bile. Push the dose below roughly 1 gram of beta-glucan per serving, as many lower-fiber instant packets do, and the gel is too thin to do meaningful work. Push the dose to 3 grams or more, which a half cup of dry steel-cut oats typically delivers, and the gel is thick enough to measurably slow gastric emptying, slow the absorption of the carbohydrates sitting alongside it, and trap a clinically meaningful fraction of bile acids across the day. This is why the dose is not a suggestion. The dose is the input that activates the entire downstream mechanism. Less than 1 gram per serving, and you are drinking flavored starch water. 3 grams per serving, and you are running a cholesterol-pulling protocol that adults on mild statin doses have used, with their doctor's supervision as a partial adjunct. The literature is consistent on this point. The dose is the lever. Now, let us take a moment to talk about what most breakfast articles skip over entirely. The blood sugar story. Because oatmeal, the way the average adult eats it, is not the gentle carb the wellness corner keeps promising. A cooked bowl contains starch, and starch, once the heat and the water have softened the crystal structure, becomes glucose almost completely available in the small intestine. The glycemic index of a food is a measure of how quickly the carbohydrate becomes blood glucose relative to pure glucose. Steel-cut oats sit at roughly 42 to 53 on the glycemic index scale. Rolled oats sit at roughly 55 to 59. Quick oats sit at roughly 66. Instant oats, the 90-second packets, sit at roughly 79 to 83. Read that sequence again. The exact same grain, processed in 4 different ways, ranges from a low glycemic load food to a high glycemic load food. The breakfast aisle does not tell you that. The packet does not tell you that. And the average adult, who has been trying to eat a healthier breakfast for a decade, is making a meaningful blood sugar decision at the grocery store shelf without knowing they are making it. Which means that the adult who picks up steel-cut oats and the adult who picks up the instant maple brown sugar packet are eating, at the molecular level, 2 genuinely different breakfasts. One produces a slow, low, durable curve of blood glucose. The other produces the same kind of sharp peak that a slice of white bread produces. Same label, different physiology, different cost to the arteries over years. Now let us connect the blood sugar story to the second molecule sitting next to the beta-glucan. The relevant compound is avenanthramide, a polyphenol unique to oats. There are at least 3 main avenanthramides in oats, called A, B, and C in the literature, and they sit in concentrations of roughly 40 to 100 parts per million in the groat. Avenanthramides are not present in wheat. They are not present in rice. They are not present in barley. They are oat-specific, and they are doing work in the body that most wellness content skips entirely. They suppress the inflammatory signaling molecule nuclear factor kappa B. They inhibit the production of inflammatory cytokines like interleukin-6 and tumor necrosis factor alpha at physiologically relevant doses. And they pair with the beta-glucan in oats to produce a documented synergistic anti-inflammatory effect, which means the soluble fiber is doing one job in the bowel and the unique polyphenol is doing another job in the bloodstream, and the 2 jobs reinforce each other. Take a moment to remember that, because the breakfast aisle is not selling those 2 molecules as a pair. The aisle sells carbohydrates. The science sells a coordinated 2-molecule anti-inflammatory breakfast. Here is the part that almost nobody talks about outside of oat science departments, the gut microbiome story. Because when beta-glucan finally arrives at the colon, undigested, The bacteria that live there begin to ferment it. The fermentation products are short-chain fatty acids, primarily propionate, butyrate, and acetate. Butyrate is the preferred fuel of the colonocytes, the cells lining your colon wall, and a well-fed colonocyte layer is a meaningful barrier against the kind of low-grade gut inflammation that has been linked in the last decade to systemic metabolic dysfunction. Propionate travels through the portal vein to the liver, where it participates in gluconeogenesis, suppresses hepatic cholesterol synthesis, and modestly improves insulin sensitivity. Acetate circulates systemically and contributes to satiety signaling through the central nervous system. The cohort studies linking whole grain oat consumption to lower LDL, lower fasting glucose, lower waist circumference, and lower systemic inflammation are now understood to be partly mediated by this axis. You are not feeding yourself when you eat a bowl of oats cooked the right way. You are feeding the bacteria that regulate your metabolism. That is the second half of why a single daily bowl has measurable effects in the literature and not on the label. Now let us address the part most wellness content treats as a problem and most honest science treats as a manageable detail. Phytic acid. Phytic acid, also called inositol hexaphosphate, is a phosphorus storage molecule in plant seeds, oats included. It binds minerals, especially zinc, iron, calcium, and magnesium, in the gut and reduces the fractional absorption of those minerals from a single meal. The popular version of this claim says phytic acid will slowly leach minerals from your bones or your blood. The honest version of the claim is narrower and more useful. Phytic acid binds dietary minerals during the meal in which you eat oats and slightly lowers the absorption percentage, particularly of iron and zinc, from that meal. It does not leach minerals you already absorbed. It does not pull calcium from your skeleton overnight. Over the course of a diverse diet, the effect is small, especially in adults who eat animal protein alongside their oats. Because the iron and zinc in animal protein come in the heme form, which phytic acid does not bind as readily. Soaking oats overnight, cooking them for longer, and pairing them with a source of vitamin C all measurably reduce the phytic acid effect of the meal. The honest position is, phytic acid is a real but minor antinutrient for the specific meal in which you eat oats, and the adults who experience meaningful iron or zinc concerns are the ones who eat oats as the bulk of their diet, not the average adult who eats one bowl a day. Now, here is the protein conversation, because most adults assume oatmeal is a small incidental protein source. The relevant number is roughly 5 grams of protein per half cup of dry rolled oats, rising to roughly 8 or 9 grams per half cup of dry steel-cut oats in some lots. That protein is roughly 15 to 20% globulin, a family distinct from wheat gluten. Oat globulin has been measured to stimulate muscle protein synthesis in older adults, including in studies from the last decade on anabolic resistance, because it has a leucine content of roughly 7 to 8% of total protein, comparable to whey. That number does not make oatmeal a complete protein source on its own. The lysine content is limiting if oats are the only protein in the diet, which is why traditional oat-based breakfasts, the ones that survived across cultures for 3,000 years, paired oats with milk, with eggs, with buttermilk, or with cheese. The mature position is, oatmeal contributes a meaningful fraction of the muscle supporting amino acids your body is asking for in the morning, and the protocol that works is the one that pairs oats with another protein source, rather than treating oats as the entire breakfast. And that pairing is exactly why every traditional oat culture, from Scottish porridge with cream to Swedish oatmeal, with milk to the German overnight oats with quark, did not treat oats as the whole meal. They treated oats as the substrate for a real protein to land on. Now, take a moment to absorb the larger picture, because the breakfast aisle has been quietly teaching a simpler story than the science actually supports. The simpler story says oats are a gentle carb, mildly heart-healthy, mildly fiber-rich, slightly better than a bagel. The science says something sharper. A measured bowl of steel-cut oats, delivering at least 3 grams of beta-glucan, paired with a real protein, free of added sugar, delivers a coordinated multi-organ input that lowers LDL through the bile acid mechanism, lowers fasting glucose through the slow starch curve, lowers systemic inflammation through the avenanthramide and NF-kappa B axis, and supports the gut microbiome through the short-chain fatty acid pathway. That is not a mild benefit. That is a coordinated whole-body input that runs in the background every single morning. The form of the grain, the dose of the soluble fiber, the absence of added sugar, the protein partner, and the polyphenol toppings. Those 5 inputs compound across months and years, and the cohort datasets are now large enough to measure the compounding. So, what does the actual science suggest you do with all of this? The protocol is 6 steps. First, pick the right form. Steel cut or thick rolled oats, not instant, not the 90-second packets. Second, measure your beta-glucan. Aim for at least 3 grams per day, which works out to roughly half a cup of dry steel-cut oats, or a slightly larger portion of rolled. Third, cook slow. 25 minutes on the stove for steel cut, which preserves more of the beta-glucan viscosity and produces the slower blood sugar curve. Fourth, skip add sugar, including honey, brown sugar, and the flavored packets, which double or triple the glycemic load of the meal in a single tablespoon. Fifth, pair with a real protein, 20 to 30 grams of Greek yogurt, a couple of eggs, whey added to the cook, cottage cheese, or any combination that brings the total protein of the meal to roughly 25 grams. Sixth, vary your toppings thoughtfully. Berries add polyphenols. Nuts add healthy fat. A small amount of cinnamon has been measured to modestly lower the postprandial glucose response of the meal. None of these 6 steps is exotic. All of them, repeated daily, are the difference between a daily bowl that does little and a daily bowl that quietly lowers your LDL, steadies your morning glucose, supports your gut microbiome, and contributes a meaningful fraction of your morning protein. The protocol is rarely a new substance. The protocol is almost always the form you choose, the dose you confirm, and the way you pair what you are already eating. Let me bring this back to where we started. 2 adults of the same age, the same cholesterol, the same fasting glucose, the same lifestyle. One eats a daily bowl of instant oats, sweetened with skim milk and a banana. The other eats a daily bowl of steel-cut oats with Greek yogurt, a handful of walnuts, and a small amount of cinnamon. 12 weeks later, the LDL numbers have separated. The fasting glucose curves have separated. The waist measurement, in the cohorts where it was tracked, has separated. The difference is not oats. Both adults thought they were eating oats. The difference is the form, the dose of beta-glucan, the absence of added sugar, the protein partner, and the toppings. That is the entire claim a careful reader of the literature will sign. And if you store only one sentence from this video about oatmeal for the rest of your life, store this one. The grain was never the variable. The form of the grain, the dose of the beta-glucan, and the bowl you actually built around it, those were the variables all along.